next week, we can come up with a study plan for each one of you. Does that sound okay? Yeah. So, guys, let's try to keep the class interactive. To the ones who've already been there, maybe you guys can help us with information that we have already covered. If we try to question you, or if you can just fill in for us, it'll be great. And as and when we go, if there's something that you know about, you can just talk, and it'll be easier and it'll be better. It'll help you recollect it better as well. So, with no further ado, I think Abhi's going to get started with B cells, and then we can move.
Yeah, hi guys. So I'll be starting off today with MHC class, the MHC molecules, and then we'll move on to the T cells and the B cells. So MHC class molecules, as you guys probably have heard, actually stands for major histocompatibility complexes, and they actually play a vital role in how our body responds to extracellular or intracellular pathogens. We'll look at each mechanism separately now. But the main thing that you have to keep in mind of this MHC proteins is that they are present on almost all cells of our body, and they are involved in even the innate immune system and the adaptive immune system. And if you can think about it, they would actually serve as like a bridge between the innate and adaptive immune system. Obviously, as a bridge, there are different T helper cells as well that go through this process, but MHC molecules actually are present in the innate immune system and the adaptive immune system. So MHC molecules actually, just a bit of history so that you can keep in mind what their function is. They were actually discovered as part of transplant medicine, and when you probably have heard when people say tissues are rejected or they are accepted. This has to do with when you transplant a foreign object into a different person's body, then our immune system will either recognize it as foreign. When it does recognize it as foreign, then it will initiate an immune response against it and they will attack it. So in that case, that tissue is considered rejected, versus acceptance is basically our immune system recognizes it, but decides that it is not something it should attack against, and that's when acceptance happens. So these MHC molecules are the main mediators or the main mechanism behind how our immune system recognizes different types of objects. So they differentiate between different types of tissues. So because over the course of evolution or over the course of years of mutations, what has happened is each MHC class molecule has a different type of allele. So they have small variations in their proteins that actually make them different. And T cells respond in a different way to one type of MHC molecule versus another type of MHC.
There's one person who's also joined the
Sorry.
meeting. Yeah, that's fine. So just to get you up to speed, Abhi's just talking about is it Aishwarya, the person joined right now?
Yeah.
Yes, it's me.
Hi, Aishwarya. So we just started a couple of minutes back. This is, I think, the second slide. We're talking about immunology today, and so the previous class we covered innate immunity, and today we're going to do adaptive immunity. Abhi's just getting started with MHC molecules. This is the first slide. At any time, any point in time, if you feel like you're left out or you don't understand what's going on, you can just leave a text, or you can just interrupt Abhi in the middle, and he'll let you know what's going on. Is that okay?
Yeah, it's okay. Yeah.
Yeah, yeah, yeah.
Yeah. So yeah, I'll just continue on with the MHC molecules. So major histocompatibility again, like I was saying, it was initially discovered in transplant medicine, and because each individual almost has a different type of, like, they have like a fingerprint of an MHC molecule, because there's small variations in the proteins that make up the MHC molecule. They can act like almost like a fingerprint. Obviously, if you have twins, or if there's some kind of special artificially created clones, then you can have similar MHC molecules. So twins, especially, it cannot be fraternal twins, they have to be identical twins. In that case, the MHC molecule of them, they are they are similar, and they can actually transplant tissues from to each other without any immune consequences. So MHC molecules, their main function is they act as membrane proteins on antigen presenting cells. So if you remember from last class, antigen presenting cells, they take up external antigens or viral antigens from intracellular pathogens, and they present it on the MHC molecules. So can any of you guys remind me from last class what type of APCs that we know, or which cells can act as antigen presenting cells?
Uh,
Macrophages?
yeah,
Yes,
and
macrophages.
dendritic cells
Yes,
and
dendritic cells.
B
Yes.
cells also. Then
Yes.
something is there in the first stage that it's Langerhans cells.
Yes, Langerhans cells. So these are different types of antigen presenting cells that are situated in different locations throughout our body, and they act as security guards. You can think of them as security guards, and they are just surveilling that whole area. And once, and suppose an microbial pathogen is met, then they will phagocytize them. Then they will go through that whole process of phagolysosome fusion, and then they will take a specific antigen and they will present it on the MHC molecule. That's what their whole name comes from, antigen presenting cells. And then eventually that can trigger different types of responses. So we last few classes we saw how that can trigger an innate immune response. In this one we'll look more towards the adaptive immune responses. So APCs, so like I said, after they digest the microbial pathogen, or it can even be a foreign body, they would display the peptide antigens. So you have to keep in mind that MHC molecules can present only peptide antigens, and peptide antigens are the ones that can be recognized by T lymphocytes. So just keep that in mind. There's an exception to it, and I'll let you know when that comes ahead. So there's another name that we call MHC molecules by, human leukocyte antigen, and it also has to do with the history because it was first recognized in transplant medicine, like I mentioned, and it was seen in the white blood cells of the transplant patients. So that's why they named it human leukocyte antigen, and it basically just means MHC. But there are specific names that you have to keep in mind because certain MHC gene variations, you can think of them as allelic variations. Those can lead to certain autoimmune diseases or some certain types of diseases these can lead to, and you have to keep those in mind. I have a list at the end, and if you went through first aid for these topics, you would know there's a small list. Um, you just have to memorize them at the end. I'll let you know about that as well. So we'll just continue on with MHC class molecules. So this is the whole process that I was mentioning. So initially the dendritic cell, which is a type of antigen presenting cell, it will capture a microbial pathogen. It will phagocytize them, it will digest them, and then it will get activated because of the digestion. During phagolysosome fusion, there are some cytokines produced. During phagocytosis, there are some cytokines produced. It will activate the dendritic cell. Some of them will end up migrating to the lymphatic tissue. Some of them will continue on to secrete cytokines to recruit further mediators of acute inflammation that we talked about last classes in the previous classes. So the ones that do end up migrating to the lymphatic nodes is what we are going to focus on mostly. So these dendritic cells, the activated ones, they end up reaching the lymphatic nodes. And if you remember from the architecture of the lymph node, there's a place that is rich in T cells, and that is situated exactly next to the afferent lymphatic vessel, meaning the ones that are going to encounter the incoming pathogens. So these T cells will then recognize these, they will have this receptor interaction. You can think of them like a handshake, and this will activate the T cells, and cause them to differentiate into different types: T helper cells and T helper cells or T regulators. It depends on the cytokines produced between these interactions, and then it will lead to an outcome, which we'll look at it. So this is the general diagram of what we're going to look at uh, in this class. So it can be T helper cell activation, and then eventually B cell activation or T cell activation, which Ashwin will talk about. So this is the general mechanism that you just need to keep in mind. We'll look at the specifics in a while. So we have MHC class 1 and class 2. So we looked at MHC. It was basically like a fingerprint for you, unless you have a twin. But yeah, you can think of it like some kind of unique identical identity. And we have two types of it: MHC class 1 and MHC class 2. Uh, so one of the main things that you have to keep in mind between MHC class 1 and 2 is the structure of them. So I know it has a lot of names. You don't have to keep a lot of them in mind, but just understand that in MHC class 1, there's something called a beta 2 microglobulin. So this specific protein is important because in diseases where MHC class 1 protein is upregulated, there is an excess of beta 2 microglobulin, and that can lead to certain diseases, specifically like some type of kidney diseases. Um, yeah, and so yeah, in MHC class 1, there's alpha and beta 2 microglobulin. In MHC class 2, it's actually identical protein, but it's alpha and beta, but they both have identical structures. Versus in MHC class 1, there's just one alpha molecule and a beta 2 microglobulin. Um, and as I mentioned, their functions are different, and also the places where MHC class 1 and class 2 are expressed are different. That has to do with their function, and I'll get into that in a bit. uh, I'll just go over some of the main reasons of why MHC provides an advantage, why we use this kind of system to in our immune system. So one of the features is that MHC, the genes that do encode for MHC molecules are polymorphic. So you might have heard this in genetics before. So a polymorphic gene is when there are uh, multiple different types of allele in the population. We'll go over the specifics, but just know that it basically means variation. So there are many variations in an MHC class molecule, and different individuals, they would express different MHC molecules. So those can also present different types of microbial peptides. So you can see the advantage over there. And another thing that you have to keep in mind is co-dominant expression. So we have 23 chromosomes from our father and 23 chromosomes from our mother. So what happens is one of the chromosome from your mother will express one type of MHC molecule, and one of the same chromosome but from your father, so the different allele, will express a different type of MHC molecule, because it would still differ between your parents. So that's why you will have different types of MHC molecules even between your siblings. But when it comes to the case of identical twins, that means the same kind of chromosome, same kind of allele is present on both chromosomes, and therefore you will not have a difference in the MHC class molecule. And that can also happen in the case of consanguinity. Yeah, so these are the different types of cells that express MHC class 1 versus class 2. So when you think of MHC class 1, you need to keep in mind that all cells, all nucleated cells, will be expressing this protein on their cell surface. That is a fact. So all nucleated cells. So that is so that means there are probably some exceptions to it. Can anyone give me an example of a cell that would probably not express MHC class 1?
PC.
Yeah. That's correct, because RBCs are not nucleated, so RBCs do not express MHC class 1. And so the thing about platelets is I had this confusion. On first aid, it has mentioned that uh, it there is MHC class 1 expression, but when I googled it, platelets also was mentioned as non-nucleated cells. So uh, I wouldn't be sure about platelets. I'll check that for you and get back to you. How about that? When Ashwin is presenting, or Ashwin, can you just check that?
Yeah, sure.
Okay. So yeah, we'll get back to you on that one. So yeah, class 1, all nucleated cells. So the only exception for now are BCs, maybe platelets, but every other nucleated cells has an MHC class 1 expression on their cell membrane. And what this means is that some of our T cells, uh, if these MHC class molecules present some of the peptides, because their main function is to present some kind of a peptide in their receptor, so that T cells can come and recognize them. So MHC class 1 usually presents to CD8 T cells. So this is called the rule of eights. It's an easy way to keep in mind. So whatever the number you multiply, you have to end up with 8. So class 1 gets CD8. So 8 times 1 is 8, versus class 2 gets 4. So 4 times 2 is 8. So that's one way you can keep in mind. So class 1 usually ends up activating CD8 positive T cells, and these are usually cytotoxic. So cytotoxic means they are toxic to the cell. So the cell that they recognize this antigen on, this MHC class 1, what happens is it would cause cellular death to that particular cell. So you can imagine what type of pathogens CD8 positive T cells would be trying to kill. It would mostly be intracellular pathogens. It would be viral pathogens, because again, viruses use our own cells to produce their machinery. So viruses, some intracellular pathogens, suppose like TB, and intracellular pathogens, and also some in this might be involved in some kind of cancer killing mechanisms as well. Versus class 2, which activates CD4, because 4 times 2 is 8, so that's the thing you have to keep in mind. Class 2 activates CD4 T helper cells, and that can end up in different types of immune responses. That can end up in antibody production, or that can end up in T cell stimulation and various other things, which we will look at. So these are the reasons why that MHC class molecule is so important to be present on your cell surface. Um, yeah, so this is just a table that I found. Just for completeness sake, I included it here, and these are the cells that have class 2 MHC molecule expression. Yeah, I forgot to mention that class 1 is all nucleated cells, but class 2 is usually present on some types of antigen presenting cells. So dendritic cells, macrophages, and some B cells. Yeah, these are the reasons why MHC molecule is expressed, and the reasons why it can be stimulated on these type of cells. I just included it. We'll go through these examples when we see them on the way. Uh, this is also a nice table I saw in Ambos. So I mentioned that MHC class 1 and MHC class 2 has different names. So the names that we call it by, because HLA-A, HLA-B, HLA-C. So if you see any of gene names, if they mention if there is a mutation, and not mutation, this patient probably has a gene that has an HLA-A3 or A2 or something like B4. If you hear those names, you should be thinking of it's a MHC class 1 molecule, versus if you hear something like DQ2, DP8, something like that, uh, basically two letters in the front, think about class 2 molecules. We know the difference. We looked at the differences between MHC class 1 and 2. One major difference is that MHC class 1 stimulates CD8 versus MHC class 2 stimulates CD4. Another difference is in their structure itself. MHC class 1 is just made of an alpha chain and a beta 2 microglobulin versus MHC class 2 is made of two symmetrical chains, but which has an alpha and beta chain. And expression wise also they are different. Expression, MHC class 1 surface of all nucleated cells, platelets. Yeah, so this is what I was talking about. In on Ambos it did say platelets have MHC class 1, so I would still go with this one. But I know where you're coming from when you say platelets do not have nuclei. uh, yeah. So MHC class 2 again only on the surface of some antigen presenting cells like dendritic cells, macrophages, and some B lymphocytes. So there is MHC class 1, which I mentioned that it reacts to intracellular pathogens and viral particles, versus MHC class 2 that mostly tries to react to extracellular microbial pathogens, usually like a bacterial attack or something like that. So there are two ways then, these antigen presenting cells. So right now we are going to be talking about how an antigen presenting cell, after eating or phagocytizing the organism, how is it going to break it down and present, how is it going to decide, is it going to go present it in the MHC class 1 or MHC class 2 protein on the cell? Because dendritic cells, antigen presenting cells, they are nucleated, so they will have MHC class 1, and they are APCs, so they will have MHC class 2. So which ones go to MHC class 1 and 2? We'll look at the exact mechanism. So one pathway is called the extracellular pathway, and this is the one that triggers MHC class 2 molecular expression on the cell surface. So extracellular pathway is MHC class 2 because it digests extracellular proteins. So what I meant by that is, suppose imagine this is a microbial antigen. Maybe this is a piece of the cell wall or something that this antigen presenting cell has phagocytized. Then once it forms the phagosome, then there's a fusion between the lysosome and the phagosome, and you have all these hydrolytic enzymes that are released that digest the full protein into peptide molecules, oligopeptides. And sorry, yeah, so you have the formation of oligopeptides. And at the same time, this phagolysosome, okay, just imagine that is happening, but at the same time your body is producing these MHC class molecules, because you need to present the digested particles to this outside cell surface. So from the nucleus you have transcription and then translation, and then you have the protein formation inside the endoplasmic reticulum. You can see this translation, and eventually with the help of some chaperone proteins you fold in the alpha and beta. So if it's alpha and beta, it's going to be MHC class 2. So what happens is, in the endoplasmic reticulum you form the MHC class 2 molecule, and you have this, suppose think of it like a uh, placeholder, because usually at this location where the alpha and beta meet together, this is where the we are supposed to present the peptide to the outside environment. But imagine in an endoplasmic reticulum there's going to be numerous proteins and numerous peptides floating around. So if this area was left open, it will just bind any one of those peptides, and it wouldn't be accurate. So it wouldn't be exactly the microbial peptide, it would just be something that was floating here. So that's why we have this kind of placeholder. It's called the invariant chain. You have in the endoplasmic reticulum, and then it goes to the Golgi for packaging and post-translational modifications. And once that happens, this vesicle, it pinches off the Golgi apparatus and joins with the phagolysosome. So in the phagolysosome, we know it's an acidic environment, and that particular environment and maybe some of the enzymes, hydrolases within the lysosome, causes the release of this invariant chain. This clip, exactly, this placeholder comes off, and now it's an active, it's an MHC molecule that is ready to bind to any peptides. So in this phagolysosome, there are digested microbial peptides floating around. One of them, if it recognizes, if it fits, it will bind to it, and once it binds, the one that is actually bound to the peptide will be moved on to the cell surface. The one that is not bound will be degraded, and then eventually this whole cycle will repeat again and again. So this is how class 2 MHC molecules are expressed. So class 2 things you have to keep in mind: extracellular pathogen, they come into phagolysosome fusion, and then eventually the peptide binds to the MHC class 2 and then expressed outside CD4, because 4 times 2 is 8. um, Next one is how MHC class 1 expression works. MHC class 1, so I told you, think when you're thinking about 1, think about intracellular pathogens and intracellular pathogens like viruses, maybe some fungi, some TB, things like that. So the viruses, they mainly made of like RNA or DNA material inside an envelope. So once they enter our cytoplasm, they use our own machinery, which is like our own protein making machinery. They use it to produce their own proteins. So the viral proteins are synthesized, but then some of these antigen presenting cells, they will they in the cytosol itself, they are able to recognize that uh, this protein is not ours, or maybe there was some mistake in the viral protein and it gets ubiquitinated. So from the first lecture, I'm from the first and second lectures, you guys remember the ubiquitin proteasome degradation pathway. So it is meant to be used to degrade intracellular proteins, basically. So once this viral protein gets tagged by this polyubiquitination, then the proteasome recognizes this tag and then starts degrading it into oligopeptides. And these oligopeptides, what happens is they get transported into the endoplasmic reticulum. So this, by the specific protein called TAP, it's called transfer associated with antigen processing. That's what TAP stands for. So what it does is, just remember that it transfers these oligopeptides into the ER. So that the MHC class 1, that is concurrently being produced and going in the ER, because our nucleus is here, it's gonna send mRNA, we are gonna produce MHC class 1 molecules. They are gonna be connected to these TAP proteins, and once these peptides are released into the endoplasmic reticulum, they are physically connected. So you can see there's a very high concentration here, so the peptides would bind more towards the MHC class 1 molecule, and then eventually they get exocytosed, and the CD8 T cells will recognize them. So yeah, it is a pretty interesting pathway how it works. Things to keep in mind are the STAP protein, so which allows the peptide to transmit into the endoplasmic reticulum so that MHC class 1 can bind it. And also you also have to keep in mind some of the differences between the pathways of MHC class 2 and 1. So 2 external, 1 internal. uh, the main difference also comes just from the structure. Beta 2 microglobulin over here you have two symmetrical alpha and beta. So the cells also, the ones that express these different types, there's a differentiation in that. Then the T cells that respond to the molecules, the MHC class 2 versus 1 is also different. 2 is CD4 and class 1 is CD8. And how do we get the protein antigen for MHC class 2 expression? MHC class 2 expression, we get it from, it's supposedly an extracellular thing that we get from the lysosome, endolysosomal protein, mostly internalized from the extracellular environment. Versus MHC class 1, it's usually a cytosolic protein. So it can be one of our own proteins that our body decides to attack against, or it can be a viral protein that is being synthesized using our own machinery. That would be in MHC class 1. And the enzymes responsible, obviously in MHC class 1, it was inside the lysosome, so it's the lysosomal proteases. In MHC class 1, the degradation actually happened in the cytosol using that ubiquitin proteasome degradation pathway. So it's the proteasome that degraded the proteins. Yeah, and also keep in mind which is the site at which the protein, the peptide gets loaded onto the MHC, because these small things, I've seen a lot of UWorld questions on this as well. So in MHC class 2, where in MHC class 2, the actual protein gets loaded onto the MHC class 2 molecule in the endolysosome, versus in MHC class 1, the protein gets loaded onto the MHC class 1 molecule in the endoplasmic reticulum. So keep that in mind. I've seen some questions on this, and it's good to remember that. And this might look like a complicated pathway, but if you think about it in terms of extracellular versus intracellular pathogens and how our body deals with them, then it makes sense, because extracellular means you have to go through phagocytosis. If it's phagocytosis, then what happens is there's phagolysosome fusion, and within the phagolysosome fusion, you have the MHC that comes in and recognizes some of the peptides. Versus if it's intracellular, you would also have some kind of phagocytosis, but that is not relevant, because the virus or the intracellular pathogen will be using our own machinery to produce protein, and we'll just degrade that, put it onto an MHC class 1, and make the CD8 cell recognize. This is just another representation of how the MHC class 1 and MHC class 2 works. If a macrophage first phagocytizes the microbe and then presents on the cell, then if CD4 T helper cells recognize it, it would cause one type of function, versus if B cell presents it, then it would cause a different type of outcome. In class 1, it's always CD8 cytotoxic T lymphocytes. It's just going to cause apoptosis of the cell in question. This is what I mentioned before. Some of the HLA subtypes, you cannot call them mutations, because they are just allelic variations that are present in the population. So maybe like 15% of Caucasians probably have B27, or so it's something along those lines. So it's not it probably developed as a mutation, but it just predisposes you to these conditions. It does, like, having this HLA subtype, which is the type of MHC molecule, doesn't mean you will get the disease, but given the right environmental factors, then you can probably develop these diseases according to the subtype. There's no easy way around this. You just have to memorize them. First aid has some good mnemonics. So B27 is pear, DQ2, DQ8, celiac disease. Yeah, you just have to memorize them. So any questions so far? Uh, mm, all right. So the next topic, Ashwin will take over. It's about T cells.
Yeah, we talked about MHC, and a lot of times we mentioned CD4, CD8. All of that can be alien terms to you right now. So, put it in context, we'll talk a little bit about T cells. It's what the MHC pairs do. And once we are done with that topic, you'll have a better idea about what Abhi was talking as well. And it'll all fit into the bigger picture. Again, same goes for this. At any time, if you feel you're lost, you can stop me and we can take on. Yeah. Can you guys see it nice and big?
Yes.
Yeah. So we'll talk about T cells. They're part of the adaptive immunity, as we call it. And adaptive basically means that instead of having a generic response, as in doing the same thing irrespective of what the pathogen is, we try to develop some kind of adaptations to better suit the pathogen. So kind of like a lock and key mechanism. We try to bring antibodies and we try to bring T cells that can recognize better and attack specific pathogen, knowing exactly what their weakness is. So it's important for cellular immunity, as in once the pathogen enters the cell and it is not in your blood anymore, there has to be mechanisms in place by which you can enter the cell yourself and destroy the pathogen. If it's out in the open, then it is easier to destroy the pathogen. That's when it's extracellular space or in the intravascular space, as in inside your blood vessels. That's when you can secrete antibodies. So antibodies are not part of the cell, right? You can secrete them into the bloodstream and they can kind of cover the pathogen, coat it with a shell of antibodies, and then you can digest them. But in case of pathogens that enter into your cell, like the mycobacterium or the viruses are basically nothing. They're not even considered living if they're outside your cell, right? Once they enter the cell is when they start living. They thrive from our resources. So to fight stuff like viruses and fungi or even mycobacteria like tuberculosis that enter your cell and attack from inside, you need cells like T cells that can invade our cells and launch an attack. There are majorly two kinds of T cells that you have to recollect. One is called helper T cells. The names are very, very helpful in remembering their actions. Cytotox, as in bat, and cytocell. So cytotoxic T cells are basically bad for the cells. They are the ones that carry out killing. They are the assassins, if you can call it. The helper T cells are kind of like the ones who arrange for it. They guide the T cells. They are the eyes and the ears. They tag the cells with which are actually the ones containing the pathogen. There are different mechanisms by which they do it, and they also T cells also help attack in pairs with B cells. So they help activate a lot of other cells in your adaptive immunity so that you can attack as a whole. They kind of again serve like a bridge, but not between innate and not just between innate and adaptive, but also between different cells in the adaptive. So every time you come across something like a CD and a number following it, this
Excuse
means
me. Your voice is the volume is kind of fluctuating. Couldn't hear properly. It, it's like sometimes it's a high pitch and sometimes low pitch. Some things we can't hear. Something like that. It's going on.
uh, I'm not sure how I can. I don't know what's the fault here, but I'm as close as to the router that I can get. If you lost me somewhere, you can tell me and then I can repeat it. It's the
Yeah,
only
this
thing
is
that I can do.
now this way we can
Oh,
hear
okay.
properly.
Okay. I think I just That's just okay. I think probably I just have to be most. Yeah. So every time you hear like we already mentioned CD8 and the other one was CD4. So what CD basically stands for is called clusters of differentiation. Again, our cells can't see; they do not have eyesight, so they have developed mechanisms by which they can recognize each other, kind of like a GPS. So we have different markers on the surface of the cells that act like signals for us to know what the cell does and what type of cell it is. So clusters of differentiation, you could say, is a kind of marker that is present on the surface of T cells and any kind of cells for that matter. And they serve like as markers. And the number that follows them is basically probably how they were discovered or the order in which they were discovered. So there's no significance to the number as such, but this is how they figured it out. So in order for us to classify T cells, we use these CD numbers as a guide. So we have cytotoxic and helper cells. Helper cells mainly contain cluster of differentiation number 4, CD4, and cytotoxic T cells mainly contain CD8. Those are the two main things you got to remember. But like I mentioned, CD4 and CD8 are specific to the exact type of T cell. But what we also need to know is that there are some markers that are common to all types of T cells, which is again important to remember. So the T cell surface markers that I mentioned in the first line, TCR, CD3, and CD28, must be there on all T cells, irrespective of what kind it is. So they are the ones that are surely there. If you want to call a cell a T cell, you have to find these markers on their surface. TCR, which is T cell receptor, CD3 cluster differentiation 3, CD28 cluster differentiation 28. You will see why CD28 and CD3 are important in maybe even the upcoming slide. So T helper cells, as we already mentioned, they have CD4. CD40 ligand and CXCR4 are again very, very important because they serve a very important function that we'll come across. So don't try to memorize this in one go. You can take your time and try to understand and memorize where the CD markers are. And it's basically root memorization. There's no logic, as I told, to how they numbered it. So you have to remember them. And cytotoxic T cells have CD8 and T regulatory cells, which basically is like the security for the security. They try to keep us from attacking our own cells. So T cells itself are the one, kind of like Abhi said, they're like security guards. They go around, and if they find any pathogens, they attack it. So there has to be a mechanism in place where we, if at all by mistake we attack a civilian, right, we attack a cell that is our own, but we we were misguided to thinking that it was a pathogen. There should be a mechanism in place for us to rectify that error. That is basically T regulator cells. Yeah, type of regulation. So you have to remember, since CD4 is already a marker for helper cells, the exact CD number that you can remember, which is very specific to regulator cells, is CD25. You might come across that in questions. It's not the most high yield information, but it's good if you can remember that. Yeah. So, like we mentioned in the previous presentation, all of the immune cells start their journey at the bone marrow, inside the bone marrow, and from there on, most of the cells go on to mature in the bone marrow itself. But there's an exception, which is the T cells, which move on to the thymus. Thymus is there in your superior mediastinum in the anterior segment, and that is where the T cells travel to from the bone marrow in order to mature. Yeah. So the big diagram on your right is the pathway that the T cell takes up until it becomes a completely mature T cell. BM progenitor cell basically stands for bone marrow progenitor cell, right? As we mentioned, bone marrow is something called a stem cell, which has extreme potential to become whatever it wants. Called plasticity. If you think about it, it's like metamorphosis. Like it is a cell with so much potential, it is not at all differentiated that it can become any type of cell it wants. From there on, we differentiate it a little more, so its functions become a little more specific. That's when we have cells like T, B, or NK progenitor cells. All of these are involved in the immune system: the T cells, B cells, and the natural killer cells. And from there on, we have something called a pro T cell, which is a little bit more specific. If you see the in the bottom denominator for every picture, there's something called DN written. So DN1, DN2, DN3, and so forth. The expansion for that is double negative. They're called double negative cells because at this point in time, neither do they have CD4 nor do they have CD8. These CD4 and CD8 are the markers that we use for T cells. So when they're developing in the bone marrow, since they do not have both of these markers, they're called double negative T cells. That's what the DN stands for. So up until now, all of this has been happening in the bone marrow. Up till the stage of late pre T cell, they're still in the bloodstream. They're traveling from the bone marrow towards the thymus. Once they reach the thymus, we looked into the histology of thymus in the last presentation, where it had a cortex and a medulla, cortex in the outside and medulla towards the inside. So once they reach the thymus, to be a little more specific, once they reach the thymic cortex is when they get all their CD markers. They get CD4 and they get CD8. So you have to recollect that once they mature, they'll have only one of these two. But initially, as and when they enter the thymus, they have both. And from then on, once they become mature, they lose one of this and they just have CD4 or CD8. But they also have the three CD markers that every T cell should have, which is CD3 and TCR for us to recollect. CD7 does not have any clinical significance, at least at our level. So you don't have to remember it. Again, there has to be a regulation in place for us to be able to differentiate our cells from the cells that are infected with the pathogen. So all the T lymphocytes go through a process of selection, initially positive selection and then negative selection before they can enter the bloodstream as mature T cells, so that they don't go rogue and attack all the cells blindly. So the first selection is called positive selection. And what happens in that is that all the T cells in the thymic cortex try to bind to the MHC class proteins. So the initial requirement for us is that the T cells have to be able to bind to the MHC in order to understand what the MHC is presenting. So MHC is like Abhi mentioned, is like a phone holder or something, right? Where you have a certain niche in which you can present the peptide or the part of the pathogen for us to recognize. So the first requirement for a T cell to do its function is that it should be able to bind to MHC. So what happens in positive selection is sorry. What happens in positive selection is we select for the lymphocytes that have the ability to bind to our MHC, and neither should it bind too tightly, neither should it bind too loosely. It has to bind to a certain extent only for it to be able to function well. So we choose only the amount of T cells that can do this function properly, and from then on, the T cells move into the medulla, and in the medulla they go undergo a process called negative selection. In this case, we in the MSC, so we have already verified the fact that these T cells are able to bind to MHC molecules, right? Now, what we do is we also have another mechanism in place where we showcase self antigens on these MHC molecules. Self antigens basically means parts of our own cells, which are physiologically needed for a human being to function. We showcase these cells on the MHC and make sure that the T cells do not react to them. That is very important, because when if that process is not done properly, then that's when we give rise to a lot of conditions called autoimmune conditions. Some of you may be familiar with the term. Autoimmune basically is when our cells attack our cells. We'll be discussing a couple of pathologies under that umbrella term in the few slides to follow. But so, in order for the cells to not react to our own cells, it is important that we first showcase these cells on the MHC and make sure that all the T cells that bind to them are neglected. So that's why it's called negative selection, because in this case, the T cells that bind are the ones who are killed. In the initial selection, it's called positive selection because the T cells that bind are the ones who are selected to move further. But in the second part, the T cells that bind to the MHC class proteins showing self antigens are the one that are killed. That's why it's called negative selection. And the exact pathway by which the T cells that are self reactive are killed is using the fast ligand pathway. So, this was one of the clinical correlations that we talked about in the second presentation, where I had mentioned that uh, we need this pathway to work. The reason why we have to remember the fast ligand pathway is because it has a clinical correlation, and this is how our T cells are selected in the time. So, if can somebody recollect how this pathway works? Maybe just a little. Some of you have been part of the initial lecture. Any recollection at all? When we were talking about apoptosis, I talked about the intrinsic pathway and the extrinsic pathway. Intrinsic was what happened inside the cell. Extrinsic was how other cells can induce apoptosis, and one of which was when a fast ligand on the cell, like a T cell, binds to a fast receptor on the cell that is going to undergo apoptosis. And the minute this happens, there is a ubiquitin that is released, and cytochrome C is activated, and there are a lot of apoptotic enzymes called the BCL2 family that helps it undergo apoptosis. I guess I answered the whole question, but I hope you guys can recollect that. If not, please just go through it again, the pathway, because this is the clinical correlation. Again, since this is a very important process, if something goes wrong in this process, we can have a lot of pathologies and a lot of autoimmune diseases. There's this protein, which is called the autoimmune regulator protein, which is responsible for showing these self antigens on the MHC. This is the protein with the function of showing these self antigens on the MHC so that the T cells can recognize, and the ones that do bind too tightly are killed. So if this protein doesn't work well, we obviously have self-reactive T cells that enter the bloodstream. And if self-reactive T cells enter the bloodstream, they will attack any which cell that they please. And there are one pathology is if AIRE protein itself is not working, we can have this condition called an autoimmune condition where there's adrenal insufficiency, chronic mucocutaneous candidiasis, and hypoparathyroidism. Adrenal insufficiency, adrenal gland is located on top of our kidneys. It is responsible for the production of a lot of steroid hormones. And if that doesn't work, we develop something we develop a condition where we do not have enough glucocorticoids, mineralocorticoids to regulate our BP. Glucocorticoids have a lot of function to do with our blood sugar level and fat deposition. So all of that can go away. Even our sex hormones, which is part of the medulla, will not be produced. Chronic mucocutaneous candidiasis is a fungal infection. Candida albicans is the pathogen that causes this infection. Mucocutaneous because it affects the layers of our mucous membranes like our mouth and also cutaneous layers like our skin. Parathyroid hormone is responsible for secreting. The parathyroid gland is responsible for secreting the parathyroid hormone, which regulates the calcium levels in our body. So, if this protein doesn't work, you can develop all three of these conditions. The exact mechanism by which this happens is not well known, so I won't dwell into that. So we have already discussed this. Abhi has also touched upon this. That's why it was important to know MHC because from that we realized that MHC class 1 showcases proteins that are intravascular, inside the cell, intracellular. That's why the major cells killed by the cytotoxic T cells are viral infected cells. So that's why you have under the area of clinical significance, you have viral infections like HIV and hepatitis B, which take over our cells mechanisms to replicate by themselves. And that's when MSC class 1 can break a part of this virus and uh, showcase it on their surface so that our cytotoxic T cells can recognize. So again, since we have reached this amount of differentiation, we have to go a little bit more into detail and discuss different types of T helper cells because they have slightly different function. Th1 is the first type that we'll discuss, and the main motive of Th1 helper cells is to promote the fight against intracellular pathogens. And the major cells that are involved in, like the major cells that are stimulated by the Th1 cells include the macrophages. And like I said, T helper cells are a bridge between different cells in the adaptive immunity itself, right? So not just the macrophages, also the cytotoxic T cells and the NK cells, and even the B cells are activated by cytokines that are released from Th1. And if Th1 doesn't function well, is when we can develop a lot of different conditions. Rheumatoid arthritis is again an autoimmune condition. Multiple sclerosis is where we attack our own myelin sheath, which is the covering that is present around our nerves. And type 1 diabetes, some of you may know, is when we produce antibodies against pancreatic islet cells. These are the cells that are responsible for secreting insulin. So if you produce T cells and B cells that are going to attack the pancreatic cells itself, you will destroy the cells that produce insulin and hence you develop diabetes. The more common diabetes that we are all aware of is type 2 diabetes. That is because of environmental factors and our diet patterns. But you can also develop type 1 diabetes at a younger age where you lose pancreatic B cells because of autoimmunity, where our own B cells and T cells start attacking the pancreatic cells. Th2 is another subtype. The most important things that you have to remember about Th2 cells is that the the other cells in adaptive immunity and innate immunity that they activate are mainly eosinophils, mast cells, and basophils. You can see the difference because in the initial case, we were trying to fight a different kind of pathogen. In this case, if you're attacked by something like a parasite, right, and helminth infections like filariasis or Wucheria bancrofti, the one that causes elephantiasis. All of these are parasites. And to fight parasites, we need secretions from cells like eosinophils, like eosinophilic basic protein. Uh, we had talked about it in the previous presentation, where these are the type of secretions that we need to fight parasitic infections. In that case, a different type of cell has to be called upon into action, right? So we have a different T helper cell called the T helper cell type 2, which helps us fight extracellular pathogens like parasites and helminths and help us launch an attack against them. When we talk about B cells, we'll realize the difference between IgG and IgE. And it is very important to remember that IgE is stimulated in type 1 hypersensitivity, which we'll talk about. And also to fight against helminthic and parasitic infections, IgE is the major type of immunoglobulin secreted by B cells. TH17 is not as high yield and well known as TH1 and TH2. TH17 also increases, it is pro inflammatory, and it increases the amount of neutrophils that have been, like, they call upon neutrophils basically to the site of action. There's a big table in, I didn't include the table in first aid because you can look at it by yourselves, where they go on to mention which are the exact cytokines that activate TH1 versus the ones that inactivate it. So you can see that the cytokines that activate TH1 will be automatically be the ones that inactivate everything else. For example, interferon gamma is important for the activation of TH1. So automatically, they also inactivate TH2 and TH17. So the ones that are present in green is activation versus the ones that are red is inactivation. So interferon gamma activates TH1. At the same time, it inhibits TH2 and TH17. On the other hand, interleukin 2 and interleukin 10, I've not mentioned it here, but interleukin 10 and interleukin 2 are important for TH2 secretion. So automatically, they inhibit the secretion of TH1 and TH17. TGF beta is kind of specific. It's actually anti-inflammatory. It stops inflammation. So, because as we know, inflammation is not a state in which our body should be at all points of time. We only need it while fighting pathogens. There has to be a mechanism in place where we know when it's time to stop and start healing. That's what TGF beta helps us with, and it's actually anti-inflammatory. So you have to, there's no other way around it. You have to remember which are the cytokines important for each function, but you can take your time with it also. You don't have to memorize it in one go. Slowly, when you learn about more pathologies, and you will help, there'll be more clinical correlations that you can think of, and you'll automatically be able to learn it better. The last type of T cells, which we already touched upon, is T regulatory cells. It promotes self tolerance, as in it prevents autoimmunity. And because of which it secretes some stuff like like we have already mentioned, TGF beta is also secreted by regulatory T cells. They also help us in anti-inflammatory actions to dull the inflammation, to actually help us heal better. Always there should be a balance, right, between pro-inflammatory and anti-inflammatory. So the ones that we looked up to up till now was pro-inflammatory, and T-regulatory cells is kind of anti-inflammatory. Again, so at the genetic level, the gene that codes for the T-regulatory cells is called FOXP3. So automatically, when the process in place for self-tolerance has an issue, we will develop autoimmune diseases. The FOXP3 deficiency is called IPEX syndrome. IPEX is actually an acronym because it stands for immune dysregulation, polyendocrinopathy, enteropathy, and since the whole disease itself is X-linked, X stands for X-linked. Immune dysregulation is very self-explanatory. Autoimmunity is because we cannot regulate the response of our T cells, and now they attack our own cells. Polyendocrinopathy is again stuff like diabetes. If you attack the cells that attack the glands that secrete hormones like insulin, you will obviously develop diabetes. Enteropathy, because the cells of the enteropathy is anything that affects your gut, that is your intestine, small and large, and your stomach, and your colon, and all of that. So anything that any highly replicating cell which can be vulnerable to the attack of T cells can be affected. So there's also we can also develop dermatologic conditions like nail dystrophy, which is dystrophy is when the cells not develop. It's an maladaptive mechanism by which the origin cells do not develop well. Dermatitis, derma is skin, itis is inflammation, so inflammation of the skin. As we already talked about, the reason why you have to remember CD40, the reason why you have to remember CD40 ligand and CD28, and why every T cell requires it is because even though we secrete these T cells into the uh, even though we secrete these T cells into the circulation after two selections, we also have another mechanism in place to help us regulate the response. Our body is pretty cautious in that manner that it has multiple steps that the T cells. It's basically like a deck of cards, and the more the amount of regulations you have in place, the more likely that it doesn't. If something goes wrong, another place is compensated, right? So there's something called so all the T cells, in order for them to be active, they need two signals, right? Not just one signal. So we have talked about MHC being one signal where the protein or the peptide or the pathogen showcased in MHC has to bind with the T cell receptor in order for the T cell to be stimulated. But that's not the only signal in place. That is signal number one, where MHC class 1 or 2, depending on that, if it's MHC 1, it'll be CD8 T cell. If it's MHC 2, it'll be CD4. So that is the first bond that takes place. The second bond that takes place is from it's between CD28 and D7. So this is again one of the place where there's some regulation. If one of these two signals don't take place, again there won't be the killing. The cytotoxic effect of T cells will not take place. This is another regulation in place, and you have to remember that B7 is present on the antigen presenting cell and CD28 is present on the T cell. And it is only when the CD28 binds with B7 and at the same time the MHC binds with the T cell receptor that the T cell can be stimulated, become active, and secrete perforins and granzymes and carry out the killing. This is so you have to remember we had positive and negative selection, and now we also have this co-stimulatory pathway, all three of which are regulations for the T cells to not attack our own cells. Again, I wanted to talk about a clinical correlation. So among the antigen presenting cells, we also have B cells. Initially, all the B cells, we will talk about it, but it is important for you to know this small snippet of information for you to understand pathology. Is that initially all B cells start up with IgM antibody on their surface. They have IgM and IgD. IgD less important, but IgM is more important. And like I had mentioned, T helper cells have the additional function of activating these B cells into their most mature kind, in which they can secrete specific antibodies depending on the antigen. So T helper cells. So initially, I mentioned T helper cells have multiple kinds, and there was TH1 and TH2. So if you want, let's say, if you want the B cell to change from IgM antibody, which is always there, to something more specific like an IgE. IgE is majorly important for allergic and helminthic infection. So let's say our body is fighting a parasite, and now we need the B cell to secrete IgE. But what is always there on a B cell is IgM. It is only when a T helper cell comes, presents the molecule, and activates the B cell by secreting different interleukin molecules that the B cell is able to recognize that, okay, IgM is not what is needed in this situation. So I have to start secreting IgE in order for us to specifically attack this antigen. So that's the mechanism by which T cells influence the secretion of antibodies from B cells. The BCR receptor is the one that recognizes the antigen. And once they receive the signal from T cells, is when they can change their expression of immunoglobulins and secrete the necessary immunoglobulins. Again, in this place, we have two signals that are needed for the activation of T cells and B cells. First is the obvious mechanism. MHC has to be uh, present the antigen to the TCR receptor. But the second signal is CD40 ligand and CD40. Here also, it's important for you to realize that CD40 ligand is the one that is there on the T cell, and CD40 is what is there on the B cell. So the reason why it's important for you to know this is because there is an actual syndrome with uh, an amount of our population is affected by the syndrome called hyper IgM syndrome, where the CD40 ligand is not present on our T cells. You can think, when you think about it as such a small defect, it's just one marker that is not there in one type of cell in your huge body. But the effect is monumental. Just because CD40 ligand is not there, the B cells are not able to convert to any other type of immunoglobulin. The only type of immunoglobulin that they can secrete is what they're born with, this IgM. So IgM cannot fight a lot of other infections that require IgE or IgG or IgD, are the different kinds of immunoglobulins that are required to fight certain type, like say parasites or bacteria with capsules that require you to have IgG. All of them cannot be all of these immunoglobulins are not made, the person with this syndrome is susceptible and vulnerable to all those infections by parasites, by different bacteria, by different viruses, because of which his lifespan is majorly affected. So hyper IgM syndrome is it's an X-linked recessive, which is also something you have to remember because the defect in this gene, the gene that codes for the CD40 ligand, is in the X chromosome. So because it's there in the X chromosome, it's an X-linked defect. And in this defect, because of the lack of CD40 ligand, the T helper cell cannot give the second signal to the B cell to convert their production of the immunoglobulin from IgM to something more specific. And hence, they cannot fight a lot of infection caused by parasites, viruses, and bacteria that require them to have a different type of immunoglobulin. It is one of the immunodeficiencies. We'll talk about it in more detail when we discuss all the immunodeficiencies, which is a very, very high yield topic for you world. It's easy points. Once we know the mechanism, then it's all very derivative and very simple to understand. So yeah, this is just one. We'll talk about a lot more, but I wanted to tell about this right here because then it'll help you remember better. So there's a pictorial representation. Normally, the MHC showcases the peptide to the T cell, and then the CD40 ligand binds to CD40. That's when the IgM is converted to something that is more that is necessary, depending on the situation, whether you need IgG, IgA, or IgE. But if that is not the case, then the only immunoglobulin that they secrete is IgM, and you have a lot of IgM, but it's not necessary. So you have something called IgM. Is that clear?
Yeah, I have a question here. As I went through that peptide, I hear that rather than the CD40L and the CD40, there's the cytokinins also the responsible for the class switching for production of.
Yeah. So how the cytokines affect is that cytokines are the ones that decide how the TH is changed, right? So initially you have a T helper cell. The decision whether it becomes TH1 or TH2 is decided by what cytokines are secreted. So they indirectly have an effect on this. So once you know what cytokine is secreted, you'll realize what type of TH cell you'll become, and from there on, you'll decide what kind of stimulation you'll give to the B cell.
Okay, thank you.
Are you guys able to follow up until now? If 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 so, you can just drop a text. Yeah. All right. I think Abhi will carry on with B cells now. Do you guys want a small break? Maybe a couple of minutes, guys.
I'm fine. I can continue.
Okay. Okay. So then probably Abhi can continue. Abhi. Yeah. I think we're pretty much left with all of you, so it's okay.
Yes, we can see. Yeah, it's clear. No, that's okay. Yeah, well understood. Shall we do some questions? That would be nice. Yeah, yeah, sure.
Guys, how about a couple of questions now? Are you both here? Sahina there? As in, like, we can do a couple of questions. You all? No, I mean, we can do 2 or 3 practice questions based on what you've covered till now. Sahina, are you back as well?
Okay, guys. So this will be the first question. Hi, guys. So this will be the first question. So just go for it and just type your answers in the chat box when you're done. You'll have like 1 minute for this. Okay, so Leah says E. Any other answers? Sahina also says E. So yeah, let's go through the question. 24. Yeah, Kusal also says E. So let's see. 24-year-old man comes to the urgent care clinic, painful swelling in his left groin. Patient noticed the swelling 2 days ago and has progressively become more tender and painful. He has no chronic medical conditions. He recently sustained a puncture wound to the sole of his left foot. So he had some kind of injury to his left foot, and now he has a swelling in his left groin. So that's painful swelling in his left groin. That's what we know so far. Physical examination shows it's an enlarged, tender, non-fluctuant. So non-fluctuant means it doesn't move around left, like move around like a balloon filled with water. So fluctuant means like a cyst filled with water, how it would move. That's what fluctuant means. And so this is not moving like that, so it's not a cyst. And left inguinal lymph node with erythematous overlying skin. So the skin on top of it is red, and the left inguinal lymph node, so that lymph node is not moving. So there's so you're right in that, in the term that it's not move, like this lymph node is like within itself it doesn't move, like it's tender, it's a solid mass because it's the lymph node that's swollen, but the lymph node itself can move. So I don't want you to confuse between cancer. So non-fluctuant doesn't have to do with how the lymph node moves in the tissue. It can move, yes, it can move, but it does, it's like a solid object, not like a rubbery kind of material. That's what non-fluctuant meant. And overlying skin, so there's a small puncture wound on the sole of the left foot which expresses pus upon mild pressure. So all signs of inflammation are present and there's some kind of acute inflammation happening. Which of the following histological findings is most likely responsible for this patient's groin mass? Atypical B cell proliferation. So atypia is usually seen in some kind of cancerous thing, and this is not cancerous. Diffuse granulomatous reaction. There's nothing here to like suggest anything to do with granulomatous reaction, and yeah, there's nothing especially concerning for granulomatous reaction over here. Lymph node necrosis, again, there is pus, so you might think necrosis, but there's nothing specific about lymph node necrosis, because what the process that happens here is acute inflammation, and within that one lymph node you might have some necrosis. It's not extensive lymph node necrosis, so you can clearly rule that out. Mark paracortical cell expansion. So paracortical area of your lymph node is usually where your T cells are. So if it's a pathogen that has to do with your T cells or something to make your T cells more proliferating, that's when you'll have paracortical cell expansion. Formation of multiple germinal centers, which every one of you guys chose, is the correct answer, because germinal centers, as we saw before, are the first the like when B cells start responding, they form primary follicles, and within them they have germinal centers. They do that antibody specificity and start priming themselves towards the antigens. So yeah, within the lymph nodes, formation of multiple germinal centers is the correct answer. Yeah, within again, so reason why it cannot be lymph node necrosis is, even though you might see some, sorry, you might see some necrosis within the inflammatory reaction, that would especially not be in the lymph node but be in the site of inflammation first thing. And lymph node necrosis, extensive, it usually happens in only specific kind of conditions. Germinal centers, you guys chose very correctly. So this is how UWorld wants you to think. Patient sustained a puncture wound and then an inflammatory reaction. So the antigen presenting cell went to the lymph nodes, and then the adaptive immune response was triggered. The B cells recognize, bind to, and process the large antigen into smaller peptides and present on the MHC class 2 receptors to the naive T cells. The naive T cells then become T helper cells, which are CD4 positive, and they secrete specific kind of cytokines to help the B cells in the germinal centers undergo somatic hypermutation and affinity maturation until they find the perfect antibodies to the pathogen. And that's why that lymph node is swollen, because that whole area is very active. Yeah. So the rest of the answers were actually caused by different specific pathogens from different diseases, but you don't need to know that. So paracortical hyperplasia, like I mentioned, usually something to do with T cells. So T cell mediated response. So in a viral infection, maybe then you might have seen paracortical hyperplasia. So learning objective, you guys got it spot on. So we're good. This is not one that we need. Yeah. Try this one. Pretty sure you guys should get it. But yeah. Yeah. Let's go through this question. So a 6-year-old boy is evaluated for recurrent infections and failure to thrive. So a young boy with recurrent infections, you should be thinking about some kind of immune system problem and immunodeficiency, something along. So some kind of immunocompromised state, something that will lead to that. You'll be thinking about that just from this sentence. Then you move on to he has been hospitalized for pneumococcal pneumonia twice, 5 episodes of otitis media requiring placement of tympanostomy tubes. So you can just take this information for face value, just elaboration of what recurrent infections is, or even something specific about them is that pneumonia is usually an infection of your respiratory tract, and respiratory tract is usually a mucosal area. So your respiratory mucosa is involved, and also your some type of your inner inner ear, middle ear is also involved. So those are some specific things you can take note of. And also history of prolonged diarrhea, so your GI mucosa is also involved. So these are the information they give, and then physical examination shows large tonsils, palpable lymph nodes, hepatosplenomegaly. Further evaluation shows that the patient has defective signaling between activated CD4 T cells and B lymphocytes. Which of the following immunoglobulins likely has the highest serum concentrations in this patient? So there are two ways you can answer this question. One, just by the fact that they have mentioned here, this patient has defective signaling between activated CD4 T cells and B lymphocytes. So as we saw before, that this usually happens, that handshake, that whole co-stimulation and everything happens, and this happens between naive T cells and mature B cells. So mature B cells are the ones that already have IgM and IgD on their cell surfaces. So as you can already know, so right now if this signaling goes wrong, the IgM and IgD would sustain, and there won't be class switching to other types of molecules. Um, so that is what you can. That's one way you can try to answer. The other way is the fact that GI mucosa is affected and the respiratory mucosa is affected. So you know that mucosal type IgA is not produced. So it can definitely not be the highest in serum concentration. And there's some inner ear defects that is still IgA or IgG because of bacterial infection. So IgA and IgG will be down. Uh, just from that fact, maybe you can try to eliminate some choices, but for this, you basically have to know that once naive T helper cells and mature B cells interact, that is what results in class switching. So initially we have IgM and IgD. So you might be wondering which one to choose. Because IgD's function is not really known, and they can be present anytime, so it's not a wise choice to choose IgD. So always go for IgM. If there's a question about which is the first antibody that's ever produced, IgM over IgD anytime. So that's why here we go with IgM. This is that syndrome that Ashwin talked about, and I showed you a picture of that baby. Um, so this is a defect in the communication. So from the T cell side there can be a defect, or from the B cell side there can be a defect, because one of them has the CD40 ligand and one of them has the CD40 receptor. So any one of them, if there's some kind of mutation in any one of these proteins, you can have hyper IgM syndrome, because IgM is initially produced and requires class switching. But in this case, there's no class switching, so IgM is the only one that accumulates. Recurrent sinopulmonary and gastrointestinal infection. So that is that one point that I tried to highlight, that diarrhea and pneumococcal pneumonias, because mucosa is usually IgA. So if you have infections in that area, you know that IgA is not being produced. So IgA is deficient. Infections with opportunistic pathogens, failure to thrive. In kids, always when they have multiple infections or something that can cause them to not have enough nutrition, so they will have failure to thrive. Failure to thrive basically, right now, just keep in mind, it means uh, a kid that cannot grow according to his or her age. That's what failure to thrive means. So that's why IgM accumulates, IgG, IgA, and IgE, because there's no class switching. Again, there's a diagnostic test called flow cytometry, in which you can detect the certain receptors or the ligands on the T or B cells, and depending on the mutation. In this case, here it says CD40 ligand that is absent on the CD4 T cell. So that is one way you can develop hyper IgM syndrome, and another way is if there is absence of the CD40 receptor on the mature B cell. That's also another way. Yeah, they just keep mentioning about the same thing. So the reason why I talked about class switching, VDJ recombination, all that stuff is, when you're reading all these UWorld answers, like the questions will not ask about that material, but the answers will always include some of those terminology. And if you have that background, you won't be lost. So right now you know that hyper IgM syndrome from a defect in class switching in B lymphocytes, and this occurs by splicing out DNA coding for different types of heavy chain constant region until the desired isotype is reached. So we saw that isotype switching, meaning the class switching from IgM to IgGA or D, it depends on the cytokines, and it's basically a change in the constant region of the heavy chain, because light chain, we saw that it only has V and a J and a small constant region, versus the heavy chain, which has a large constant region that induces macrophage and complement activation. Also has has its own properties that is what determines its class type. So you need to change that in isotype switching. Yeah, so you can have CD40 ligand deficiency, which is the most common X-linked recessive, or the uncommon one is the CD40 deficiency on the B cell. It's autosomal recessive, but will lead to the same disease because once this handshake doesn't happen, there's no class switching, so you cannot respond to all the other different types of pathogens that these guys do the function for. That's basically it, hyper IgM syndrome. It's a nice clinical correlation, and yeah, so I think yeah, Ashwin will take over right now.
Okay, so there's one more question based on immunoglobulins. Do you guys want to do that right now, or move ahead and come back to it later?
Shall we do it now?
Yeah, go. Yeah, sure. That's pretty straightforward, I guess. But we can still get. Yeah. So there's A, B, C, D, E. It's just that you can't see it. What do you say, guys? You have Sahana's answer. How about Leah? Yeah, so there are options from A to E. Cool, we just check. Right. So the question was the immune response observed in an apparently healthy 12-year-old Caucasian male after recurrent exposure to bacterial antigens is characterized by a rapid increase in serum IgG level. Some immunoglobulin molecules are attached to the surface of macrophages, neutrophils, and B lymphocytes. Which of the following is the cell attachment site for the immunoglobulin molecule shown on the slide below? So basically, what they're trying to tell is on recurrent exposure, as in there has been one exposure which has helped us to create adaptive immunity, and now we are producing serum IgG, which is specific for that antigen. And the way this serum IgG molecule is working is by attaching itself to cells like macrophages, neutrophils, and B lymphocytes to help with the killing. So they're asking which part of the immunoglobulin shown is the part that is responsible for attaching to all these molecules, which is macrophages, neutrophils, and B lymphocytes. All of you answered E, which is the correct answer. So basically, this is B had shown you the diagram. There's the heavy chain and there's the light chain, and the constant region, which determines which type of antibody it's going to be, is determined by the constant region. And in the constant region, there are two sites. One is both of which are important because there are mechanisms by which these immunoglobulins kill. These immunoglobulins usually bind to the structure and then the opposite end. So they bind using the attachment at this region, the one that's shown in green, the variable region, and then they bind to another cell using the constant region. And that cell is what secretes the enzymes like granzymes or porphyrins or lysozymes and all those enzymes to kill the pathogen. So it binds to the pathogen from one end, the green end, and binds to the cell responsible for the killing with the other end. And the exact answer was E, which is the last part of the constant region, which all of both of you got correctly. The basic IgG molecule is pictured here. and identification. Yeah. Receptors. So they bind to the receptors on macrophages, neutrophils, and B lymphocytes. So every time they say FC region, they basically mean constant region. The C in FC stands for constant region. The whole process by which these immunoglobulins coat a pathogen surface and then help us digest the pathogen is called opsonization, right? Just a fancy $1 million word, but what it just means is that these antibodies coat the pathogen and using the FC region. So the macrophages cannot bind to the pathogen. You need these antibodies to recognize the pathogen, bind using the FAB region, which is more specific than anything else, and then using the FC region bind to the macrophage so that then the macrophage can digest whatever is bound to the immunoglobulin. The whole process is called opsonization. We'll come across it later on. They go ahead to describe what all the regions are. So you can just look into this diagram. Uh, the green region is responsible for binding to the pathogen or the antigen. Uh, the D region in the question, which is the initial part of the constant region, a heavy chain, is responsible for binding to complement. Next, the proteins that we're going to discuss next is the complement pathway, and you'll realize that one of the ways in which these immunoglobulins can kill is also by the activation of complements. So, if the complements are the proteins that are going to do the killing, the binding takes place in this region. Option number D. Sorry, option number D, and E is where the macrophages bind. That is all I had to show last this question. Both of you got it right. Just pass the presentation. Yeah, so complements. So blood is made up of RBCs and plasma, and in the plasma we have a lot of other proteins present in the plasma. One of which is the complements. They're also part of the innate immunity. They're part of the innate immunity because there's nothing specific about their response. They just respond to anything and everything in one of the manners, in few of the manners in which they know, irrespective of what attacks. So they're part of the innate immune system. And just as we saw right now, how the immunoglobulins have an FC region for binding to cells like macrophages, they also had a region to bind to complements. So the method by which antibodies kill is one of the methods by which antibodies kill is by letting these antibodies bind to complement pathways, bind to complement proteins, and hence do the killing. We'll see how the complements do the killing. So they're usually very small proteins, and they're there in their precursors form in the blood. As in, all the proteins should be in a certain structure, in a certain shape for it to be active. So if there is no threat, if there's no pathogen, there's no need for these complement pathways to be active, and hence they're in their precursor form. It is only when there is a trigger that they change their active pathway. And depending on what is the trigger, there are three different pathways that the complements can take for themselves to be activated. And there are the names of the three pathways are classical pathway, alternate pathway, and Manners binding pathway. So, how important it is to know every step in this pathway is debatable. It's not that important, but it's good to have a general idea because it'll help. So, there are different conditions in which we check for different levels of like let's complement factor 3 or complement factor 4. You'll realize why we do that if you know the pathway. So just be familiar with the pathway. There's only a single page in the FA, so I won't take a lot of time with it. What you have to remember is the pathways differ according to the trigger, and the trigger for the activation of the complement pathway, classical complement pathway, is an antigen antibody complex. So when a complement, did somebody so when a complement comes in close contact with an antigen and an antibody complex. Basically, an antigen attached to an antibody. When a complement comes in contact with it, it can activate a pathway known as the classic pathway. On the other hand, there's this pathway that is happening spontaneously in the background, in which there's hydrolysis of the precursor protein to form active forms of the complement proteins. But this pathway, although it is spontaneous hydrolysis, there are other mechanisms which make sure that these pathways are only activated when necessary, which we'll come across shortly. And then the last pathway is mannose binding lecithin. Mannose is basically sugar, right? It's a sugar molecule with a 5 carbon chain. And they are usually present in the wall of the bacteria or different pathogens. So, on recognition of these mannose binding lecithins on the wall of the bacteria, like basically sugar on coating the microbes, we activate the mannose binding lecithin pathway, which is very similar to the classic pathway. And once they are activated, what happens is we form a final component called the MAC, which is a membrane attack complex. And this is the complex which does the killing in the complement pathway. So what it does is that different complement proteins get together and form sort of like a channel, and then they puncture the cell. And when you puncture the cell, you disrupt the homeostasis. The different ions that are present on both sides of the membrane, the different amount of water that are present on both sides of the membrane are disrupted. And when you puncture it, all the water, depending on the gradient, will go in or out, because of which the cell will die. Or it can also lead to activating of macrophages, because there are certain complements that are dual function. Not only do they activate the MAC, they also have other functions. When I show you the pathway, you'll have a better understanding. And there are certain complement proteins with very specific functions that you have to be aware of, because there's a lot of clinical significance. So the classic pathway and the alternate pathway are the two most important pathways. We'll start with the classic pathway. It's nice to contrast between the two while studying, so that way you'll get done with both of them at the same time and help you remember both of them better. So, like I mentioned, the classic pathway is triggered by coming into contact with an antigen, antigen and an antibody complex. The first step towards the beginning of the picture is when a microbe is there. There are antigens on the surface of the microbe and an immunoglobulin, in this case IgG. So usually it's IgG or IgM that binds to the complement, and these immunoglobulin and antigen and antibody complex activate C1, which is the first in the cascade of the complement pathway. And this C1 is obviously in a precursor form. This C1 is then, what C1 does is acts as an enzyme for the cleavage of two other complement proteins, which is C4 and C2. So C1 cleaves C4 to form C4A and C4B, which is basically A and B is based on the size. The bigger one is called A, if I'm not wrong, and the smaller one is called B. So the C1 acts as an enzyme for C4 to be converted to C4A and B, and likewise for C2 to be converted to C2A and C2B. Once that happens, the antigen antibody complex plus C4B and C2B come together to form a complex, right? Stay with me. The antigen and the antibody complex activated C1, which acted as an enzyme for the conversion of C4 and C2 to active compounds, which is C4A and C4B and C2A and C2B. Then C4B and C2B attached with the antigen antibody complex to form a whole big component. This component is called C3 convertase. So all the pathways will eventually lead to the formation of something called a C3 convertase, which will lead to further reactions, and the end will produce MAC, which is membrane activation complex. So this is how the classic pathway forms the C3 convertase. And the C3 convertase in classic pathway is comprised of C4B and C2B, and the antigen and antibody complex, obviously. Then the next step, by far the most important component in the whole among all the complement protein, is C3. After this complex is formed, the formation of this complex activates the conversion of C3 to C3A and C3B. And then C3B attaches to this complex to form the final convertase, which is called the C5 convertase, which has C4B, C2B from before, and then now C3B has also been attached to this. This whole convertase is capable of activating MAC, which is membrane attack complex. As you can see towards the bottom of this diagram, so we have C4B, C2B, and 3B. Then they go on to activate C5, then C6, C7, C8, all the way up to C9, and 6, 7, 8 attach with 9 to form MAC. MAC is membrane activating complex, and the function of this is to form a pore on the membrane of the cell, thereby creating a osmotic disbalance and leading to the death of the cell. So the basic function of the classic pathway is to form the MAC, and one way to do it is using so the basic function of the complement pathway is to form the MAC, and the one way to do it is classic pathway. The other way to do it is alternate. As we discussed, the trigger for alternate pathway is spontaneous lysis of C3 to become C3A and C3B. So in this pathway, we needed C4 and C2 to be activated in order for them to cleave C3. But in this pathway, C3 is cleaved by itself. And we are continuously producing C3A and C3B. Then we have something called factor D and factor B. This is where the specificity comes in this pathway. Factor D, okay. No, okay. Are you getting this idea from how I presented it in this one? No. So, but in this slide, I think the way I presented it might have confused you, but all three pathways should end up in the formation of MAC. The reason all these three pathways exist is to end up in MAC formation. I just put it up at the end because so initially there are three, and then all of them lead to just one. So three, and then there's just one, which is MAC formation. All of them lead to the same end output. So the alternate pathway continuously lyses C3 to C3A and C3B, but the specificity in this pathway arises because the you only have factor B and factor D activation upon contact with PAMs and DAMs, which is pathogen-associated molecular patterns and damage-associated molecular patterns. We learned it in the first lecture where we said this is how our receptors get to know that there's inflammation or there's a pathogen around. So yeah, so factor B and factor D gets activated in their presence, and after the spontaneous lysis, the C3B attaches to a component of factor B called big B small b. So it's very derivative. There's factor B and a small. It gets cleaved into A and B. So now you have big B small b. I'm pointing at it with the cursor. So we have C3B, which was spontaneously formed, and in the presence of factor D, factor B is cleaved into big B small b, and that attaches with C3B. Again, it's not that important to know the specifics, but just remember this is how it happens. And then it attaches to another molecule of C3B and it forms its C5 convertase. So basically, we want to get to C5 convertase. The way we did it in classic pathway is by including C1, 4, and C2. In this one, we have just C3B and factor B. We have formed the C5 convertase, and that will help us form the MAC complex, and hence we'll do the killing. In the lectin pathway, the trigger is different. The trigger in the lectin pathway is basically the mannose binding lectin that is found on the surface of bacteria, pathogens like bacteria, and that activates two proteases called MASP1 and MASP2, which can then cleave C4 to C4A and C4B and C2 into C2A and C2B, which is very similar to the classic pathway. In classic pathway, we needed the antigen antibody complex. The only difference between that and the mannose binding lectin pathway is that in this pathway, we do not need the antigen antibody complex. All we need is the surface of the pathogen to have MBL, which has serine proteases, which help us in cleaving 4 and 2. So from this point on, it is exactly similar to this. Once we get the C3 convertase, we form the C5 convertase in the exact same manner, and from then on, we form the MAC. All you have to know is there are three pathways, three different triggers. Remember the triggers, and remember that it ends up in the formation of C3 convertase, C5 convertase, and then it goes on to form MAC, which is important for us to kill. Now for the important functions, this is what you have to remember, is that although we had C3A and C3B and only C3B was involved in this pathway, the components that are left out, the bigger components or the smaller ones, the C3Bs or the C2As and C4As all have functions. That is how, so I initially mentioned either the complement activation can lead to MAC formation or macrophage or neutrophil activation. So the way they found MAC, we have already discussed, but the way they activate other cells and the way how the other functions that they do is is based on how these components function. So C3b is important for the process of opsonization, which, like we've mentioned before, is the process by which you coat a bacteria with components that you can recognize by your cells. Like your cells will not have the lock and key mechanism for a certain antigen, but they will have a key to the lock, which is the component protein. So if they have a structure that is that fits the complement protein, if the complement protein is binding to something else, that can also be digested with the complement protein. The complement protein in the form of C3b binds to the pathogen, and just like how immunoglobulins could activate macrophages to do the killing, C3b also can activate macrophages, because macrophages have receptors for C3b, and then they'll be digested and killed. So C3b, just like immunoglobulin G, is a helps with the process of opsonization. C3A, C4A, and C5A, which were not involved in the formation of MAC, are called anaphylotoxins. Some of you might have come across the word anaphylaxis. Anaphylaxis is basically a type of shock in which, yeah, it's a extremely severe systemic allergic reaction, if you can say so. It activates IgE. IgE secretes a histamine, because of which is vasodilation, bronchoconstriction. You find it hard to breathe. Your tissues are swelling up, and you have an anaphylactic shock. So C3A, C4A, and C5A are capable of inducing anaphylaxis by activating mast cells, basophils, and eosinophils. C5A also does another additional function of chemotaxis of neutrophils, which is also an important. Can anybody recall what are the two other components that we learned that are important for neutrophil chemotaxis? There were two, one interleukin and one leukotriene that was important. Okay, it was interleukin 8 and leukotriene B4.
People, yeah.
Right. So with that, add C5A also, and these three are really important for neutrophil chemotaxis. So automatically, when you have deficiencies of the early complement pathway, early complement proteins like the C1 to C5, you have problems with forming the MAC and clearing antibodies, because they are important for the process of opsonization. You need C3b to help you clear the microbe and antigen antibody complex, the one that was the initial trigger for the classic pathway to begin. If the complement proteins are deficient and you cannot recognize this complex and you cannot clear it, you can lead to a lot of diseases. Some of you might be familiar with the term lupus, which is an extremely common autoimmune condition, and it's a very devastating condition as well, because it affects systemically, and a lot of your organs are affected. So if in people who are deficient in this initial complement factors, they are more prone to developing systemic lupus erythematosus, and they also get a lot of other common infections, pyogenic infections that form pus, basically, and also glomerulonephritis, which is we have the glomerulus in the kidney, and inflammation of which is called glomerulonephritis. And because in kidney there's this process of filtration. There's this process of filtration in the kidneys, and it's very porous. So if you have antigens and antibody complexes accumulating in the bloodstream, they will go clog the pores in the glomerulus, and it can cause inflammation of the glomerulus. And you can have antigen and antibody complexes depositing in the glomerulus, like big clumps. We'll talk about it. So when that happens, you can have inflammation of the glomerulus, which is called glomerulonephritis. An important, extremely important clinical correlation is that this was related to the deficiency in the initial complement pathways. But if the latter complement pathways, that is C5 to C9, is deficient, you cannot form MAC. And when you cannot form MAC, you're extremely prone to a specific infection caused by Neisseria, the bacterial species of Neisseria, which is Neisseria gonorrhea and Neisseria meningitidis. And you're extremely susceptible to infection by these two in particular. There's another one that I found out that so Obviously, if you have these complement pathways being randomly activated, just like T cells and B cells, it is not good for us. So we have to have find a way for keep the complement pathway in control, for which we have different proteins and different components. One of one of those proteins is called factor H, and when you're deficient in that, you can develop a syndrome called hemolytic uremic syndrome, which again is a syndrome that comprises of bleeding complications and anemia and also kidney failure. We'll talk about it when we do hemonc, but just remember this correlation. It's just for you to understand how important the complement pathway can be. Another condition that can that you can come across, which is related to this pathway, is called hereditary angioedema. So it's an autosomal dominant condition. You know what that means. Autosomal, as in it's not part of the X or the Y chromosome, the gene that encodes this deficient protein, and it also dominant, as in you only need one of your alleles to have this mutation for you to develop this infection, for you to develop this condition. So hereditary angioedema, as in your vessels are leaky and they're edematous, as in they leak a lot of plasma proteins into your extracellular space, and it can cause swelling. So one of the common conditions in which we know this happens is anaphylaxis, where you have vasodilation because of histamines, and it happens. But in that case, you have not only do you have swelling, you also have the irresistible urge to itch, right? And all over your skin, it's very erythematous and itchy. Like it's very red, it's very itchy, and it is swollen. So the important clinical differentiation between anaphylaxis and angioedema is that when you have swelling and puffiness of your skin because of angioedema, there is no itching involved. You have angioedema without urticaria. Urticaria is the sensation to itch, and erythema of the skin. So you have swelling, the skin is swollen, but it is not itchy and it is not red. And this process can take place anywhere. It can take place in your face, your mouth, or your extremities, and even your stomach. And if it takes place in the inside of your intestines, it can cause diarrhea and other problems. Oh wait, I did not tell you what is deficient. So in this condition of hereditary angioedema, the we were discussing about regulatory proteins for the complement pathway, and that's when I mentioned factor H. The second regulatory protein we have is called C1 esterase. It's mentioned here. C1 esterase is important for degrading C1. So as you know, C1 is important to initiate the classical pathway in complement cascade. But if so, C1 esterase is important to degrade C1 so that we don't activate the classical pathway unnecessarily. But if that doesn't, if we do not have C1 esterase, we automatically start activating the pathway unnecessarily, and it can lead to downward complications. But what we also need to know for us to understand the pathophysiology of this condition is that not only does C1 esterase degrade C1, but it also has a few other important functions. One towards your left, this one is it's basically part of the coagulation cascade. Even the coagulation cascade is basically how we activate different factors to form good clots, like coagulation, as in blood coagulation, as in formation, clot formation, and we have different factors for it, like factor 12 to factor 1. So to for the activation of factor 12 to its active form, that is 12A and 11A and so on, you need the C1 you need you need C1 esterase to prevent this activation as well, just like how it prevents the activation of C1. So if you lack the C1 esterase, you can cause improper activation of the coagulation cascade, just like you cause improper activation of the complement. Not just that, it can also prevent formation of kallikrein. Kallikrein is important for formation. It has a downstream effect, which is it helps in the formation of bradykinin, which we discussed in the first lecture as a mediator. Is that bradykinin, just kind of like histamine, causes vasodilation, causes increased permeability, and also it was important in the sensation of pain, along with prostaglandin E2. So, not only does C1 esterase prevent the formation of active form of C1 and prevents the formation of the active form of the coagulation cascade factors, but it also prevents kallikrein, which is important for the formation of bradykinin. So since the patients with hereditary angioedema do not have C1 esterase, they cannot prevent the unnecessary formation of bradykinin, and hence more bradykinin is produced, and hence you have vasodilation, which is how you get the swelling, which is how you get the angioedema. More vasodilation and endothelial contraction, because of which there are more space between your blood vessel cells coating, and there's more plasma proteins and fluid leaking out, and hence more swelling. So that's how you get angioedema in this condition. C1 esterase not only does it improperly activate the coagulation cascade and the complement pathway, it also uh, it also causes increased accumulation of bradykinin, which is responsible for the pathophysiology of hereditary angioedema. Another clinical correlation that you have to remember is because of ACE inhibitors. ACE inhibitors also ACE it's an it's a drug that we use to treat hypertension. It's called ACE inhibitors. The common ones are called captopril, and any drug that ends with pril, IL, enalapril, captopril. All of them are ACE inhibitors. It is a class of drugs that we use to treat hypertension. What you have to know is that the enzyme inhibited by ACE inhibitors, which is the ACE enzyme, angiotensin converting enzyme, also has an effect on the production of bradykinin. So, for patients with hereditary angioedema who are already prone to increase bradykinin, if you also give them ACE inhibitors, you inhibit the only other mechanism that is one of the other mechanisms that is in place for them to regulate bradykinin production. So since they're already producing more bradykinin than they should, if you give them ACE, you just push them over the edge, and now they're producing way too much bradykinin. So one of the most common symptoms for this, one of the most common triggers, medical triggers for hereditary angioedema is ACE inhibitors. In a patient who has hypertension, so up until then he didn't have any manifestations of the disease. But as soon as you start giving ACE inhibitors, he starts developing these, the swelling and all the manifestations of hereditary angioedema. So stroke, I'm not. So he does. So if you're asking if stroke can be a complication of giving ACE inhibitors, then no. I'm not. Oh, okay, okay. Yeah, all right. Yeah. Yeah. So you have to remember how ACE can also increase bradykinin in patients with hereditary angioedema. So in those patients, you should not give ACE inhibitors. They are one of the first line drugs that you give for hypertension, and hypertension is a very common illness. So you have to be extremely careful to not give them ACE inhibitors if they have hereditary angioedema in their family. Uh, and also you have to remember, Laya was familiar with the term anaphylaxis, and the most common drug that we use to treat that is epinephrine. Epinephrine also has an effect on our vessels. It helps us vasoconstrict and reverse the effects of histamine and many other mediators that are seen in anaphylaxis. So that is a drug that we use to treat it. But it wouldn't work in a condition like this, because the main reason here is not histamine, is not an anaphylactic mediator, is because of bradykinin. So we have to using epinephrine does not help us in this condition. This might not seem that important to you now, but the fact is that anaphylaxis is a very life-threatening condition, and the first thing that you see as soon as get a patient who is swollen all over and is itchy and red is you give them a shot of epinephrine. You don't take a lot of time before you do that. So you have to realize that once you do that and the patient is not getting any better, there is a possibility that this patient is not having an allergic reaction. Rather, he's having a condition called hereditary angioedema. So in this case, the medications that you will treat are stuff that are antagonist. So that's why people with allergies carry EpiPens, because as soon as you get an allergic reaction, you have to use epinephrine to reverse it. But in this case, it wouldn't help because it's a different pathophysiology. And in this case, the drugs that help are antagonistic drugs. That is, drugs that have an opposite effect on the same receptor. Of, like suppose there's a bradykinin receptor, you have a drug that has an opposite effect when it binds to the receptors of bradykinin, which is it is human synthesis. man-made synthesized drugs like icatibant and ecallantide, which helps prevent the formation of bradykinin. So they'll help us fight hereditary angioedema. Yeah, this is the swelling I was talking about. As you can see, so if the swelling was like an allergic reaction, there would be a lot of itching, and the whole area might be red. But in this case, that's not happening. Even in this one, so the swelling is progressively increasing up to 24 to 72 hours. It can keep increasing. This is the patient at 0 hour, and she slowly becomes worse up until she develops complete swelling. And this is a really important condition because this can block her airways, and she can have difficulty breathing and die. So the first thing that you should do in this such a patient is try to stabilize their airway. Another condition that is pretty beautifully fits into this whole concept is called paroxysmal nocturnal hemoglobinuria. In this condition, we have a defect in a gene that codes for GPI anchors. So GPI anchors are basically well, it's an anchor that helps us attach these regulatory proteins to the cell membrane, so that so when complement comes along attacking every other cell, you need to have a regulatory pathway in place in the membrane so that they realize, okay, this cell is not under attack and it's our own cell and we shouldn't attack, just like any other immune regulator that we have that we've discussed up until now. So we have stuff like DAF, decay activating factor, and membrane inhibitor of reactive lysis, MIR. So these are again identified with clusters of differentiation CD55 and CD59. We have them on the membrane. Usually in a normal human being, you have them on their membrane, and then when the complements come along and they see CD55 and 59, they realize that, okay, these cells are not to be harmed. But when you have a gene defect that helps us code for anchors, which helps place these CD55 and 59 on the membrane, when you have a defect there, the anchors are not formed, and these two molecules, 55 and 59, are not anchored onto the membrane. And therefore, when the complement comes along, they do not have anything to stop them from getting activated, and hence there's intravascular hemolysis, as in inside the blood vessel itself, they can these RBCs or WBCs or RBCs and platelets and all that is there in the blood vessel can undergo rupture because of formation of MAC or activation of other neutrophils or macrophages. So since this defect is in the stem cell, all the cells that are produced in the bone marrow, like WBCs, RBCs, and platelets, can be affected. So the symptoms are very derivative. If you have a defect in the amount of RBCs, you develop anemia. If you have a problem with platelets, you can develop thrombosis or bleeding. So if thrombosis develops in the brain, it's called cerebral venous thrombosis. In the so there are two circulations in the liver. It can affect the hepatic vein. That's called Budd-Chiari. And if it affects the portal vein, well, it's called portal vein thrombosis. And then there is also erectile dysfunction and abdominal pain, which is part of the symptoms, which is because once continuously you have intravascular hemolysis, hemoglobin is running free in the blood vessels. And this hemoglobin captures the nitric oxide and all the other vasodilators, which can cause spasm of smooth muscles. So when you have spasms in the smooth muscles, you can have abdominal pain and all the other symptoms that is seen in paroxysmal nocturnal hemoglobinuria. And because it's a mutation, it can also lead to leukemia, which is a type of cancer of the blood. The diagnosis, this is why clusters of differentiation are important, because we have developed a mechanism by which we can identify these clusters of differentiation on the surface. It's called flow cytometry, where we tag protein, we tag cells, and based on what they are showcasing on their membrane, we realize what kind of cell it is. So flow cytometry, if you use it on a patient with this defect, you will realize that this patient lacks cells that can showcase CD55 and 59. So therefore, you can realize that the diagnosis is paroxysmal hemoglobin, paroxysmal nocturnal hemoglobinuria. And even the treatment for this condition is very, very relevant to what we just learned. It's called eculizumab. It is a human-made antibody, and what it inhibits, the antibody is against C5, which is complement factor 5. So as we know, all the C3 convertase and the C5 convertase ends up activating C5 complement to form MAC. So if you inhibit the formation of MAC, you inhibit complement causing hemolysis. So I mean, basically the RBCs and the platelets are not affected. So therefore, all the devastating effects of PNH can be avoided. So the treatment for this is eculizumab, which is a human-made antibody against complement factor 5. So this is a table that you have to learn on your own. Like a lot of other stuff that we discussed today, this is also you have to commit it to memory. But when you go through this presentation, just when you learn this table, put extra emphasis on the all the highlighted stuff. I've highlighted stuff that are more important than the other information. So try to memorize them better and also go through everything on this page. And if you find this helpful, use the mnemonic. A hot T-bone steak is the mnemonics there on that's there on first aid, and it'll help you remember it better. Right. Even this page, it's so we. I think we can just run through this. So I've already told that all T cells, some T cells have more CD markers, different CD markers from the other, but some CD markers are common to all of them. TCR, CD28, which is required for the co-stimulation, and CD3, which is part of the TCR complex, is there on all T cells. But in specific, helper T cells will have CD4, cytotoxic T cells will have CD8, regulatory T cells will have CD25. B cells, they have to respond to the T cells, so they have CD40. They have MHC2 because they are antigen presenting cells. They have B7, which is the one that binds to CD28. So all these three are explainable. We have BC3, BCR, which is B cell receptor. CD19, 20, and 21, which is something you have to commit to memory. These are the three CDs that are commonly seen in B cells. So if there's a question and they say that according to flow cytometry, the cells seen in the experiment express CD21, you have to realize that the cells that they're talking about is B cells. CD21 is especially important because there is a pathogen called the Epstein-Barr virus that enters our cells using the CD21 as a receptor. So you have to remember CD21. Yeah, so just go through this page and pay extra emphasis on the ones that are highlighted. CD14 is very specific for macrophages, so you have to remember that. Likely CD16 and 50, likewise CD16 and 56 are specific for NK cells, so you have to remember that. Next, we'll talk about hypersensitivity reactions. All of this up until now was very theoretical. Hypersensitivity is completely important for has a lot of clinical correlations. So basically, hypersensitivity means hyper, like you can understand from the word itself. It's excessive reaction to a pathogenic process. So every time the immune response is every time there's an immune response, it should be adequate. It should be good enough to allergies. So sometimes you use the words interchangeably, hypersensitivity and allergies, but they're not the same word. One type of hypersensitivity is an allergy, but there are three other types that we'll talk about today that you have to realize is not an allergy, but is also an excessive reaction to a pathogen. So, like I said, when there's a pathogen, you have to adequately react to it. You shouldn't over. So imagine, suppose there's a bacteria, you fight it with neutrophils, but then you just keep producing neutrophils and macrophages, and they keep secreting more and more cytokines, and the whole inflammatory pathway is exceedingly activated. So you'll kill the pathogen, but in the process there'll be a lot of collateral damage. A lot of the cells that are part of our system that we need for our survival will be affected. So basically, hypersensitivity is a term given to any excessive pathogenic immune response. To it can be either to a foreign antigen or it can even happen to a self antigen. There are four types. The first three types involve antibodies, and the last type involves T cells. It's important. Type 4 hypersensitivity is caused by T cells, and the first three are somehow related to antibodies. This is a brief summary of all of them, but we'll just go through all of them in detail. So the first one, like I mentioned, is type 1 hypersensitivity, which is synonymous to allergies, like Leah has already mentioned. You can get allergic to pretty much everything. One of the most common ones, the nuts in food or animal sources. One sec, I think I've put on the. Yeah, so basically, no, the the big guns in the food that you have to remember is nuts, eggs, and soy. In animal sources, any bee sting or wasp or cats or insects. Environmental factors, most commonly and the most devastating, is dust, because it's pretty much everywhere. And if you're allergic to dust, it can really screw you over, because dust is ubiquitous. It's everywhere. You can be allergic to latex, like the material that condoms and gloves are made of. You can be allergic to pollen and mold. Atopic diseases basically include allergic. So you can be allergic and you can react differently. You can have so if the major reaction is in your lungs, you can develop a response called asthma. Or if it's in your eyes, you can have allergic conjunctivitis. If it's in your upper respiratory tract, you can have allergic rhinitis, which is basically your nose, pharynx, and oropharynx and nasopharynx being affected. You can even have dermatitis, where your skin is the major organ that is affected. And there are many medications also that can cause allergies, mainly antibiotics, penicillin, all of them can also cause. Like I mentioned before, IgE is the most important antibody when it comes to allergies, because IgE is the one that uh, the cells that mediate allergies have receptors for IgE. So the cells that we're talking about is mast cells, basophils, and eosinophils. These cells have a receptor for IgE, and therefore IgE is the only type of antibody that can instigate an allergic reaction. Because only when IgE cluster together and bind to a mast cell, they then degranulate towards the right. They then degranulate and they secrete a lot of enzymes like leukotrienes and prostaglandins. These are the ones that cause the symptoms of an allergy, which is edema, redness, which is erythema, and itching. Like I had mentioned in angioedema, there is edema, which is similar, but the most characteristic finding in an allergy, which both of you will be familiar with, is pruritus, which is itchiness, and also how red the tissue becomes, erythema. Like we mentioned, it's mediated by IgE, and the effect is very, very acute. As soon as you're in contact with an allergen, you can develop it. People choke on their food, like one when they're eating shellfish in a restaurant, and all of a sudden they have an anaphylactic. That's why they have to carry an EpiPen, because there's not enough time for you to go to the hospital and then get checked by a doctor. Between that gap, you can have an anaphylactic shock that is potentially life-threatening. So since the effect is very acute, you have to carry medication with you. In this case, an epinephrine pen. So there are, although the effect that we know of is very acute, there can also be a effect that is prolonged, that is delayed. The initial effect is mediated by histamines and tryptase and leukotrienes, which is basically the edema, redness, and itching. But there can also be an effect which lasts longer, which is mediated by eosinophils, where there's release of eosinophilic basic protein and there's local inflammation and damage. But that happens a few hours later. What you have, the takeaway points is that the major mediator in this is mast cells, basophils, and eosinophils, and major cells are the mast cells, basophils, and eosinophils, and the major enzymes or the mediators secreted are histamines, leukotrienes. And tryptase is important because it is very specific for mast cell degranulation, and it is the enzyme that we measure in clinics for us to know whether the mast cells are active or not. The enzyme that we measure is tryptase, so you have to remember it specifically. This is an example of allergic reactions. The left one is pretty, most of you are familiar with it. The in the middle is allergic conjunctivitis, and the right one is called hive. The process is called dermatographism, where so basically it's touch mediated. The minute you touch, mechanically, if you touch the person's skin, and suppose you write something, that trauma itself is enough to trigger a mast cell degranulation and edema and all that. So the process is called dermatographism, and in this case, it's called hives. And the patient just went on to write hives, just with your hand or with the back end of a pencil. If you just write on the person's skin, he can develop an immune reaction to that, and he can, have this phenomenon. Are all hypersensitivities congenital? No. It's not congenital. You can develop it. You can develop it because of problems with your own immune system that you develop through the course of your life. You can become allergic to something at the age of 30 or at the age of 40, and it is not. All of them are not congenital. And the next one is type 2 hypersensitivity. Forget about IgE. Other ones are important in this case. So in this case, what happens is there's an antigen that is expressed on the cell surface, which is rigid. It is not moving in the bloodstream. It is a rigid cell surface that is expressing an antigen, and we start producing antibodies to it. And when that happens, there's of antigen antibody complex, which can either the antigen can, like if the antibody is an IgG, with the other free end, it can bind to something like a macrophage and cause death, or it can bind to complement and cause death. Basically, you have antibody mediated cellular toxicity, as in the free end of the antibody can bind to one of the cells that can do the killing. So what you have to remember is that obviously you have to remember the conditions. There are a lot of examples, and you have to remember, if possible, the exact segment to which we develop the antibody to. In this case, immune thrombocytopenia, immune as in it's mediated by our own immune system. Thrombocytopenia as in thrombocyte is a platelet, penia as in deficiency. So we have platelet deficiency because we develop antibodies against platelets. And platelets usually they have projections on their cell surface, one of which is called GP2B3A. It's important once we learn about homeostasis and how primary clot and secondary clot is formed, you'll realize why GP2B3A is important. And if you develop this condition called immune thrombocytopenia, is when our body develops antibodies against this particular antigen. So if they see this particular antigen on the surface of a platelet, the antibody goes and attaches to it, and then there's inflammation of the cells because of all the cells that you've mentioned for macrophages or components and all of that. Then there's something called hemolytic disease of the newborn, which is another beautifully fitting example. So again, our RBCs also have a lot of projections on their surface. Basically, it's sugar molecules, carbs basically, and they form different structures. That's how we get ABO blood grouping. A is a type of structure, B is a type of structure, O is the absence of any structure. And not just AB blood grouping, we also have other antigens on the RBCs, one of which is called the RH factor. This is the rhesus factor. So there are some part of the population that is RH positive, as in they have RH on their RBC, some that are not. So the ones that are not, if they somehow develop antibodies to ones that are, suppose you have a person who's RH negative, and you give blood that has RH positive antibodies in it. That will cause these antibodies to bind to the. I'm sorry. I think I made a. I think I made a small error. Yeah. So in RH we have one of two kinds. It is RH positive and RH negative. So what happens is, in an RH negative person, since I'm sorry, I lost track of the RH part. Abhi, are you here? Yeah, do. Mhm. Look at this. There's group A, group B, group AB, and O, which we are familiar with, different types of blood groups. So what happens is, in a person with group A blood group, there's an RBC which is coated with group A on their surface. Different food that we consume or different microorganisms that enter our body through our gut can somehow get us exposed to B. But since B is not a self-antigen, as in is not something that we produce, our body reacts to it like a foreign substance and produces antibodies against, which is called anti-B antibodies. So in our blood, we have RBCs which have group A. I'm taking the example of a person who has a blood group A. So in our blood, we have RBCs which have group A, and in our bloodstream, we'll have antibodies against the other types. That is why you cannot, when you transfuse blood, a person with a blood group can only transfuse blood to another person with the same blood group. You cannot transfuse a person with blood group B can transfuse blood to a person with A, because he'll have antibodies to the opposite, the one that he's not presenting on his RBC. So if you're group A and exposed to B, you'll develop antibodies against it. So in your blood, you have antibodies against B. So if you somehow receive blood from a patient of a different blood group, these antibodies will bind to the RBCs and cause hemolysis. Now, this is regarding the main blood groups, but not just that. We also have other antigens on our RBCs. One family is called rhesus family, which is rhesus factor, RH factor. And the population is divided into two: RH positive and RH negative. So RH positive is the exact. There's one certain type of RH phenotype we consider as RH positive, and these people. Uh, so when an RH negative person receives blood from a person who's Rh positive, he'll get stimulated to develop antibodies against the Rh factor. And the next time he comes in contact, the antibodies that he produces will attach to the RBCs that have Rh on their surface and cause hemolysis. That is what happens in a newborn. So in a newborn, if the let's say the mother is Rh negative, but the kid, the firstborn kid, is Rh positive. So in that case, when their blood mixes, the mother is exposed to the Rh antigen and she recognizes it as foreign and produces antibodies to it. And once she produces antibodies to it, and she has the next kid who's also Rh positive, who's having this Rh factor on the RBCs, the antibodies that he produced initially will bind to these RBCs and cause hemolysis, which is called hemolytic disease of the newborn. I'm sorry that I got confused a little bit there, but I hope it's clear now. Yeah, transfusion reactions I already mentioned. You cannot mix and match between different blood groups because you produce antibodies to the blood group that you're not showcasing on your RBC. Goodpasture syndrome is a condition which affects the lungs and your kidneys. In this condition, you develop antibodies against a certain type of collagen called as type 4 collagen, which is there in your basement membrane. And so, if once you start making antibodies to this collagen type, it's an autoimmune condition. So you affect your own cells, and because of which, if it affects the lungs, you can have a symptom called hemoptysis, where you start spitting out blood because there's inflammation in your lungs because of this, and you neutrophils and macrophages come and digest tissue in your lungs, and thereby blood is leaking into your alveoli, and you start coughing up blood. It can happen in your kidneys, and you can start peeing red because you start peeing hemoglobin. Myasthenia gravis, some of you might be familiar with this condition as well, is the major neurotransmitter between the nerve cells that stimulate our muscles is called acetylcholine. It's the motor neurons communicate between themselves by secreting acetylcholine and receiving it. If you develop an antibody that is opposite in shape to a receptor of acetylcholine, that's when your muscle cells cannot communicate, cannot be stimulated. That's when you have symptoms like droopy eyelids because you cannot open your eyelid because you cannot pass the signal from your brain to your eyes for you to open your eyelid. And you have different symptoms like muscle fatigue, drooping eyelids, or and so on and on. But it's derivative of what I just said. So in this example of myasthenia gravis, you had an antibody that when it binds to the receptor, it stops the downstream processes, right? It stops acetylcholine receptor was bound to by this antibody, and it stopped the acetylcholine from doing its function. On the other hand, in Graves' disease, you have an antibody that mimics thyrotropin. The thyrotropin is secreted in order for you to stimulate production of more thyroxine, more thyroid hormones, which is secreted by the pituitary gland, but it is a regulated process. So when you have enough, it doesn't secrete more. But if you develop antibodies that can bypass this and stimulate the thyrotropin by itself, then you have unregulated production of thyroxine, and hence you have all the symptoms of Graves' disease. Pemphigus vulgaris is another skin condition. Desmoglein is a protein that is there in one of the in your surfaces in your skin. There's a point. Desmoglein is a protein that is shown on the cell, which helps it with binding with other cells and has a few cytoskeletal functions. So when you develop antibodies towards it, there is cell lysis, adherolysis, and the communication between the different layers of your skin is interrupted. So you form bullae, you form blisters, easily pop, and it's a devastating condition. So I have photos of different conditions. To the left top, you have Graves' disease, a very specific condition where a lot of extracellular matrix is deposited in the eyes. So the patient has bulging eyes, which is a common symptom of Graves' disease. It is a condition to produce more thyroxine. In the middle, you have hemolytic disease of the newborn, where because the newborn's RBCs were showcasing Rh antigen, and antibodies are made by the mother against it, and there was hemolysis. There was more vascular permeability, and all the different organs in the body underwent damage, and the kid is born like this edematous and ruddy appearance because of all the hemolysis. The the four pictures of the drooping eyelids is of myasthenia gravis, is when the patient is instructed to close the eyes, but one of the eyelids doesn't open back up because you cannot signal the eyelid to open because the neurotransmitter that you use for motor neurons is acetylcholine, and that is lacking. The last photo is that of uh, there's a photo that right now. Oh, okay, that was so the one is the newborn, where the oxygen incompatibility happens, and the baby is all edematous and red because of the hemolysis and the end organ damage. And then the one below is myasthenia gravis, and to the right is pemphigus vulgaris, where desmoglein is targeted by the antibody, and you have a lot of bullae. It's called when you have big blisters filled with stuff. Yeah, the third one is type 3 hypersensitivity. Difference is there and antibodies are already formed and are circulating in your bloodstream, and it is randomly processed in different parts of the body. So automatically, because this process is already in the bloodstream, the effect of this condition is a lot of issues affected by the same. And there's a different activity and there's inflammation. Either you die by night or you have anything. You already touched upon SLE. One ways by which you can develop SLE was when you have deficiency of another possible and another so it's also pathophysiological disease, one of them. The second one is this. So all bled of the cancers before they grade. So all antibodies, so that's RNA, they never come in contact with antibodies. But there's a problem with clearing the apoptic bodies. Apoptic bodies perfect chance for the T cells and the B cells to use DNA. So if there's a problem with these bodies and your DNA RNA leaving your cells get exposed, so in SLE, you have antibodies. You have your own DNA. So that is not used like your own. And that's why a lot of respirators, all these and even very specific all those aspects. The body has the antibodies and antibodies which are formed when the T cells and hence arthritis, which is very have very interesting. So in this condition, we develop antibodies against an antibody. So right now we have an IgM, which we enter the antibodies and forms like a cube. So we form antibodies. We form IgG and binds to antibodies. We form antibodies that into the IgM antibody, and which is and that's how we have there's a bacteria, and the normal manifestation of the disease. Different things of the bacteria. It looks similar to something on the cell of the body, and their body come in contact with the nephrons, mistake it to be the strains of the bacteria, and they start attacking our kidneys, and hence we develop glomerulonephritis. Then there's also another reaction, which more than it having chemical reasons, very specific, like type 3 hypersensitivity can be explained very well by these two. So when you give immune modulating agents, we talk about a lot of antibodies up until now, human-made antibodies, right? Like eculizumab and all that. And we also have antibodies that we derive from animals and we use the lab. All of these, although they are antibodies, they can be recognized as foreign antigens. The antibody itself can be recognized as foreign antigens, and our body can have reaction. That is what is called serum sickness, and it can also happen with vaccinations. That's when we have like some of the adversity that vaccinations face is because some of the vaccinations can kind of be rejected by the body, sort of as foreign. That's a challenge by making vaccinations to fool our body into not reacting to the vaccine itself. And vaccines can also be treated as foreign objects, and we can have a reaction to that in the same manner of type 3 hypersensitivity. We have a complex of an antigen and an antibody. As in, we form antibodies to that vaccine compound, and we form a complex, and that triggers inflammation. Polyarthritis models is a type of vasculitis, an inflammation of the vessels, and in which you have a lot of fibrinoid necrosis that are discussed in this lecture, where you have a lot of fibrinoid necrosis that attaches the walls of your vessels, and it can cause inflammation in different organs in your body. I have a picture for SLE. SLE, the pathognomonic rash, very specific rash from malar rash. It covers your nasolabial, it covers your malar region below your eyelid. And like I mentioned, SLE has a lot of clinical manifestations. Then there is glomerulonephritis. There are three kinds of imaging. One is the electron microscope, light microscope, and immunofluorescence. It just shows you the complexes that have been accumulated. All the green that you see is complexes of antigen antibodies that have accumulated or deposited in the glomerulus. Then this, as I mentioned, arthritis, and this is a manifestation of rheumatoid arthritis as a symptom in a patient. Type 4 hypersensitivity. So this is the only hypersensitivity that involves T cells. In this case, what happens is, so I had mentioned how T cells get activated. We have antigen presenting cells that show a molecule on the MHC, and the MHC, depending on which MHC it is, different T cells activate. This is the very important question in the book. It's because of poison ivy. So when people are fighting or their legs are exposed, become a man called develop contact that, and when that speaks of and speaks of blisters, there's a clinical implication to this as well. Usually to know if patients have Tito's or not, which is a protein that we get from the we get that. We have TB response to TB, and because majorly interest is so consequences to that. So if they have this protein before, there's no swelling. Most of the before, and then you have to that's called BB test or man test. And there are a lot of criteria that can be, so at least swelling, for whom like a even if it's not swelling, there are criteria for this being important. So basically, on this, new setup, just like which I do, which I get at China. Yeah, and it wouldn't matter. Like even if like so so the problem is if not this way, like supposed to be like stuff like that. So the patient who's receiving blood and he's up in the like one age group, there's some you can see anyway. And so when you try antibody so they react antibody is not used for not reactive, that can also possible. And there are 2 months of or conditions so it's been that develop the foreign body therefore his things and because these and energy. So like something that something clinical is it okay now? Yes, that's me. Yeah. If I can just that's not if I'm going to do yeah, so energy and not so within the policy we talk about the idea and I think Dublin so there's some talks the anaphylactic reaction to this protein is also a lot. So since the amount of protein is a lot, the anaphylactic reaction is also a reaction, and also left to like like IgA is a reaction. That means a reaction will feel. So next is there are a lot of there are WBCs and RBCs when you all of them but the and all these once you cancer president they look them like have I want to look that causes fever and they are meant to both body in these stuff that fight the fever like you prevent this condition when the first fever is activated inactivated so that can't get each so we need so this is avoided. Both these are comfortable, but these. So if not much there is a process in place, like you is a process to WB, and the but must not be not hence you go about this is everything. So the thing was able to stand in, and if everybody has video, the FS also so you have you have hypertension. You have because knees so the knees are used because that is an anatomical location, and because like this becomes red, you have touching all of these are very rare. Not because it helps for you and all the way from so you have a shock in this condition. What is preventable is all you have to do is before you give into and cross-matching you get and give a sample and chicken that you and there's a reaction. And if there's a reaction, then you get so might as well service. But if there's Robin, so the process we get the service. But when some like it's a basic but you realize that and manage it easily take a bit of assessment you and see there's any and so don't it's called transfusion associated and transfusion associated the firm CRA license. You have to test who is receiving samples and so there is women who have been pregnant or anyone who can have who is has been exposed so if I work with blood transfusion I don't have WBs so because that is I have antibodies so the next time I do in my blood there are antibodies to different WBCs and so if you have ever received blood you can treat them and they don't name blood. I did these can trigger neutrophils in the vesicle and process blood key and body, which will recognize and cause them. When this happens in the lungs, like the endothelium of the and in the whole dimension for where and related to expression, these neutrophils in the alveoli, they cause called edema. And the respiratory and the person can obviously respiratory to be the first with oxygen instead of intake and usually happens out of for fresh food type of plastic or factors usually get more. So when with those situations like cancer, you and getting neutrophils in the patient, you have to realize don't already need the antigen is the one who influence that. In case of food, is usually you've donated to so much is not for your vascular and both conditions you can't. That's why it is one because these are values like develop and to from them and therefore having quality because I don't know and it's hard to find so there are some that you can so you take an ultrasound see if the is it is not able to card and then realize TA was lost. But if I'm having to examine the patient completely realize then it's acute kidney injury. This is like a marker of the marker of heart. So if the heart damage and everything didn't be by you can see on the face. But this was new to and so there's a curated inside the lens of a white fiction and you see the movement. This is a photo of fire usually only animals caused by a car collision having a face. There's also the one that having into this in the sushi being happened in the not much being use of yeah B yeah